tight junctions. And there are some proteins, transmembrane proteins there, by means of which they are opposed, and they have different names like occludins, claudins, junctional adhesive molecule, or in this way. They are different names and they have some important functions. They may allow small molecules like water, some ions or solutes to pass through, semi-permeable. It makes the membrane semi-permeable. Suppose it is renal tubule and it is extracellular space, interstitial space, lumen and epithelial cells, semi-permeable membrane almost. They will allow water, ions and small molecules to pass, and they may be selective in some cases, and their tightness varies. Suppose in case of GI system, upper part of the small intestine and jejunum and ileum, they are more leaky, and in the colon it is to some extent more tight. Permeability varies. And there are numerous proteins in this way. They almost obliterate the space. It is called tight junction or zonula occludens. And its important function is that it will prevent the transport of molecules from this side, the basal side, to the apical side, or from the apical side to the basal side. That is, transporters or other proteins, those have their presence in the apical side, have no entry to the basal side. On the other hand, the proteins embedded in the basal side, they cannot diffuse or move through this to the apical side. In this way, they maintain polarity, and it is and for this, the movement of specific element is possible. Suppose sodium potassium ATPase is present in this way, sodium pump, and it is maintaining low sodium concentration here, and the apical side is a transporter of glucose and sodium. So by this way, transport can occur. This transporter has the capacity to carry glucose and sodium from the luminal side into the cell. But this protein is selectively present in the luminal side, maintains polarity. And when it enters glucose and sodium is pushed by this, and glucose can diffuse, and separate types of transporter may bring it into the interstitial space to be carried by the blood vessel. Secondary active transport can possible, and it is the function of the tight junctions. Here, tight junction. They are present in the epithelial cells, membrane of the epithelial cells of GI system, and also renal system, choroid plexus also. They maintain the polarity and they are selectively permeable. In some portion it is relatively leaky. In proximal tubule it is more leaky than the distal nephron. Leaky. They can allow the molecules to pass. And recent editions, they say that they have other also important functions. They get information from intracellular proteins. So they have some information so that they may proliferate, and metabolic functions including movement or so may be there. Proliferation may be there. But primarily it is the structure for this type of different polarity and maintaining the fluid. There is a movement of this, and through the cell, and also in this paracellular route. Paracellular route. It is called paracellular route, and it is specific in some cases, to ions in some cases. Specific. Some ion to pass through. And there are different types of proteins, occludins, clodins, and JM or this type of molecules. And they may be related with some diseases also. Leakiness varies. It is tight junction. And below the tight junction is the belt junction or jonular adherence. Jonular adherence. Below this is they come together, and there are some proteins, and they are connected to filaments, microfilaments. Jonular adherence. At the basal side, suppose GI tract, renal epithelium, this type of structure is there. It is called belt junctions. They almost surround like belt, this cell. Belt junctions, and they are called jonular adherents. And they are attached to microfilaments within the cells. And there are some transmembrane proteins. And they also contain catenanes. Catenanes. And here you are seeing the single profile. In other part neighboring cells, they are connected by other junctions, spot junctions or desmosomes. Desmosomes. Suppose one cell is connected by a patch with a neighboring cell. It is called desmosomes. And there are some proteins that are disposed in these desmosomes. Catenanes mainly. And there are intermediate filaments. Intermediate filaments. They are disposed in this way. Intermediate filaments. Desmosome. They are spot junction, patches. Rivet kora jemo, rivet. R-E-V-I-V-E-T. Rivet. Rivet like structures. And in this way, they cause a strong connection between the cells. Desmosomes. And within the cells, these catenanes or other proteins, they are connected with the intermediate filaments. Intermediate filaments. In case of jonular adherence, this is the microfilament, and they are different in structure than in the intermediate filaments. Intermediate filaments. They are different structure. Yeah. And they provide this strong support. They do not allow the cells to separate. So they provide the mechanical junctions. They provide these mechanical junctions mainly. And there are within the cells. But outside the cells, there is the basal lamina. Basal lamina. There are some junctions at the basal lamina, neighboring part. And they are composed of hemidesmosomes. Hemidesmosomes. And focal adhesions. Hemidesmosomes. Half desmosomes. Suppose in this case, there are some transmembrane proteins, and there is the intermediate filaments. But in the basal side, it is not so. So it is the hemidesmosomes. They are the intermediate filaments. But this proteins is not cadherin as in the spot junctions or desmosomes. And they are integrins variety. It is called hemidesmosomes. It is connected with the some filament-like structures, proteins in the basement, laminar basement membrane. Basement membrane. They have different names, I cannot remember in detail. It is called hemidesmosomes, half desmosomes. and in some cases there are focal junctions and is related to movement of the cells. Suppose epithelial cell, suppose it is epithelial cell of a capillary and it is the lumen, and a cell may attach temporarily, and here there are the integrin like structures, integrin within the cell and the actin filament in this, and when they bind with it by molecular motors, and it is the point of fixation and from which there is distortion of the cell and there is movement. In this way they can move. It is called focal adhesions. Integrins are there in the cells and they unite by means of cell adhesion molecules of the neighboring epithelium, and there are microfilaments or actin filaments within, and it forms a point of fixation from which there by means of molecular weight there will be shape of this change in the shape of the cell and it can move, and this is called focal adhesions. And this movement is required physiologically, and in this way they can find the space in between the cells and they can then migrate. Suppose in this condition they can feel, and there are other cell adhesion molecules and they can pass through this and they can come out from the lumen to the outside. It's called focal adhesions. And more important is the gap junction, and they provide mechanical support to some extent, but there are gap junctions and it is important for the examination, and it will not give the mechanical support, but this movement of some substances from one cell to another without entering the extracellular fluid. Normally this space between the cell, neighboring cell is 25 nanometer, and it is reduced in some cases to 3 nanometer or so. And here there are some proteins, they are disposed in such a way they form the tunnel like structure, closely opposed to one another, and these tunnels are formed of proteins, and they together they are called the connections. And it is formed of the subunits. 6 subunits, they are called connexins. If you now enlarge this space, you will find that this type of structure is there. One subunit, it is another subunit on one side, it is on the other side. Almost obliterate this space. And there is a tunnel through which it is the cell, it is cell membrane, and it is the space in between the cells, and it is reduced to very small so that it can enter through and through without entering into the cell. And it is the one subunit of one connection, and they are called connexins. Suppose they are disposed in this way. These subunits are called connections. If you take this face and disposed in this way, and it is the lumen, and it is the connexins, and they oppose one another. They are lined in this way, so that through this tunnel, substrate small molecules can pass through. Approximately 2 nanometer in diameter, and they will allow water and small molecules up to 1000 kilodaltons. That is this, they can pass through. Ions, sugars, amino acids, they can allow. And they are present in many tissues. Importantly, in the cardiac tissue, heart muscle, they are present. Visceral type of smooth muscle, they are present. And they are also present in other tissues also. Within the brain, liver, they are present. And small molecules can pass through without entering into the extracellular fluid. But the nature has the capacity of variation according to the requirement. It does not mean that always they will go from one cell to another. There are some areas where there may be one channel on one side, but not on the other side. So it can come into the ECM also, neighboring. And nature has made this facility so that small molecules can pass from the cytosol to the extracellular fluid according to the requirement. There is variations in the nature. Yeah. So, and there may be channel in one cell, not corresponding channel in the opposite cell. And in this case, small molecules can pass from the cell into the extracellular fluid. And it is so. And they are not, it appears that they are always remain in isolated conditions. Good numbers, clusters are present. Good numbers are present in a particular site. You will find in the textbooks there are different areas, and they contain neighboring this type of structure. They are all connections, and they are all communicate. Some areas, not isolated in all cases. In this way, this gap. And these connections are important structures. And gap junctions has a role like electrical activity can pass from one cell to another. Suppose in the ventricular muscle, if there is an apparent focus of, there is the action potential, and it will spread slowly, it will to all the muscle cells, In a visceral type of smooth muscle, if one smooth muscle cell gets activated by means of gap junctions, all the cells that are connected to one another in this way, they all get their electrical activity. It is the benefit of the tissues that all the cells does not require its private innervation. If one cell is stimulated, it will gradually involve all the neighboring cells. Where the time is not so much important, slowly it can involve. So individual cells does not require its private innervation. In skeletal muscle cell, every cell has its individual innervation. But in this visceral type of smooth muscle, all the cells does not require innervation. Action potential generated in a cell for depolarization, it will spread through these gap junctions. And it is related to development, and also some diseases are linked to these connections. 20 genes are responsible to produce these connections, and their structure differs in different sites. Charcot-Marie-Tooth disease and X-linked disease of peripheral neuropathy is linked to this connection. Some skin diseases like Clauston syndrome or so, they are linked to these connections. Connections. Some degenerative brain disease, cardiac disease, liver disease, they are linked to these connections. So these are these gap junctions. It is for the, in this way, small molecules up to the molecular 1000 kilodalton, they can, molecular mass, they can traverse through this. And they are also not passive conduits. And in physiological conditions, like ions or electrical charge, and they can move together and they may increase or decrease the permeability of this type of channels. Yeah. So it is these intercellular connections. In your common text, they give the basis, and you may take the help of other books and internet to have detailed knowledge. Good number of diseases are linked to this, and their causation and other particulars are given in detail in internet. It is not possible for me to remember everything. It is left for you to have your additional knowledge. Now we come to this fiber-like structure, the microfilament or intermediate filament we have discussed, and they are linked to the cytoskeleton. Within our bodies, there are the skeleton by means of which we are maintaining our structure and we are moving by different muscles. Similarly, within the cells, there are structures, fiber-like structures that maintain its shape and also allows its movement by means of molecular motors. The fiber-like structures are of three varieties. There is the microtubule, microfilaments, and intermediate filaments. Microtubules, they are hollow tubules. Only by the electron microscope they can be identified. And microtubules are present in almost all cells, and they are hollow tubes formed of the alpha and beta tubulins. Alpha and beta tubulins. Suppose in this way, in this, if you take a cross section, 13, 14 subunits is there. The alpha, beta, in this way there are 30s, and this is 5 nanometer, and it is 15, another 5 nanometer. Their dimension, lumen. They are formed of alpha and beta tubulins. Alpha and beta tubulins. Initially, there is formation of a sheet-like structures, and there is folding, and we are getting this. And the cross section there is 13 in this way. And the dimension is like, there is the wall is the 5 nanometer, lumen 15, and it is 25. They are diameter. They are hollow structures. hollow structures, alpha and beta tubulins, and they are associated whenever and they are produced from centrosome, and here there are gamma tubulin, and it is an active process, GTP, or there is these binding proteins, and there are other proteins by means of which they are produced from these centrosomes, gamma tubules, and in this way microtubules are produced from these gamma tubules. They are from the gamma tubulin they are produced, elongated, they are hollow structures. In this way, there is a hollow. On one side, there is formation, and another side is their dissociation, assembly side and disassembly side. It is the assembly side and it is the disassembly side. Disassembly, and it is called plus n, and it is called this minus n. In this way, they are produced. Yeah. And they help in many ways. We have all learned about the mitosis. In school days, the cells do proliferate. Initially is the division of the nucleus. That is called karyokinesis. And ultimately, this cytoplasmic portion or the whole cells, they divide. And in this way, two identical cells are produced. And spindle formation is there, mitotic spindle. And mitotic spindle is formed by this microtubules. So they provide the tracts by means of which they can be separated. Chromosomes can be separated. Microtubules. And near the nucleus of all the dividing cells, they have a structure called centrosome. Near the nucleus of a cell, there is a structure right angle to one another, and it is called centrosomes. And the centrosomes are formed of this microtubule-like structures. There is 9 at the wall. Regular array is there. Suppose it is in this way, and all in the 9, and there are 9 triplets. In this. They are situated 1 to 9. And they are at right angles to one another. And during the cell division, before the cell division, they will also divide by some means. And they go to opposite pole of the nucleus. Opposite pole of the nucleus. And they are connected by this microtubule. And the nucleus break down, and these chromosomes, they unite with it. And they will move in this way. You can remember these stages: prophase, metaphase, anaphase, telophase. And ultimately they reach this. And then cytoplasm will do divide. So we had there is the role of this microtubules and molecular motor cells. And they also provide the tracts by means of which proteins, vesicles, small molecules, mitochondria can go from one side of the cell to another. They will provide the tracks. With the help of molecular motors, they can do this function. And they have this plus end and minus end. Some molecular motor like kinesin move this cargo to this plus end and dynein to the minus end in general in this molecular motor. They move the cargo to this plus end and also the minus end. And by means of different drugs, their movement can be restricted. Colchicine can bind with the microtubules, and in that case, the cells, and then the process cannot take place properly, and these chromosomes can be identified. Vinblastine, an anti-neoplastic drug that can paclitaxel or Taxol, they somehow have some effect so that the mitosis is arrested. Anti-cancer drug in this. Many things are given in the text. I cannot remember everything in details. It is the basic part that I can remember. Extempo. Now microfilaments. What are they? And dimensions are relatively smaller, 4 to 6 nanometer in diameter. Microfilaments are formed of actin molecules. They are also rounded structure. এবং এই মাইক্রোটিউবুল কি করে তৈরি হচ্ছে? পুঁতি আছে না, পুঁতির কাজ যারা জানে তারা জানে। পুঁতি প্রথমে একটা দিয়ে করে একটা ডিফারেন্ট কালার দিয়ে, হয়তো নেবারিং কালার একটা শীট তৈরি হলো, তারপর শীটটাকে মুড়ে দিলে পরেই তোমার একটা টানেল তৈরি হয়ে গেল। সেইভাবেই নেচার এখানে তৈরি করছে। It is an active process. Numerous proteins are there, and some drugs can have effect over this, and diseases can have effect on these proteins. There are gamma tubulins at the beginning, and there is the assembly and the disassembly. And actin filaments are similar structures, and they have formed of individual spherical proteins. Actin, G-actin, globular actin, and they combine one with another to form the F-actin or fibrous actin. G-actin may be soluble, but fibrous actin is insoluble. And they are disposed in this way. In this double helical structure and dimension, suppose 6 nanometer, actin molecules, they come together and they produce this type of total structure is, and they are formed of nanometer dimension, but the length is much more, μm dimensions. In skeletal muscle, when you will read, then they are in the regular array, 2 to 2.2 μm or their dimension length, thin filament. But it is present in almost all the cells. Cellular movement is required. They are connected to some cell membrane by means of intermediary proteins. Suppose in RBC membrane, there are this type of structures that are called glycophorins, and by means of some adapter molecules and this actin filament is bound by means of some adapter molecules. If they are bound, they can be bound. In textbook you will find these structures, and so by means of molecular motor they can change the shape of the cell. And in case of skeletal muscle, by means of a molecular motor like myosin, different forms of myosin, it can has the power of contraction, movement of this molecular motor, and they are called myosins. They are of different classes, 18 classes or so. And within all the cells they are present. Actin is one of the important proteins in the living body. 15% of the protein altogether may be due to this actin. And they are conserved. And there is similarity between the rabbit and the yeast actin in the textbook says. 80% their common mode of their amino acid sequence, highly conserved. Actin filaments, microfilaments. And I discussed in case of cell addition along with cell addition molecules or so, they are connected in intercellular connections. Is in focal additions and weld junctions, they are microfilaments. Focal addition is a site from which the shape can be moved with the help of myosin. Yeah. In the epithelial cells of intestine, where are there microvilli? Within the microvilli, there is this type of structure. And by means of molecular motor, they can move, and the movement of the villi is possible. Or microvilli is possible by means of this structure. Yeah. Now, intermediate filaments. They are present in almost all the cells. They are the structural molecules. They are rod-like structures. They are also formed of subunits. And the subunits, they are disposed. You would have to take the help of biochemistry text or internet. They are disposed in such a way. They are also tetramer. They are formed of, they are tetramers. And this is the N-terminal portion of the cell, N-terminal. And it is the C-terminal part of this individual protofilament. And then there is another, this type of N-terminal protein. There is the structure, central is the rod part, C-terminal. But in this case, it is attached with C-terminal. N-terminal. Antiparallel. In this way, they are disposed. N-terminal and C-terminal protofilament. And it is united with this, and it is rotated in this way. Dorir moto pakiye dilo. And another two in the antiparallel. Left side C-terminal, right side N-terminal. And they are twisted together. And they are disposed in this way. And they form one structure. They together form one structure, one filament. And then there is another filament in this way, but at some distance. And when multiple of this filaments is produced, and they have some periodicity. And in this way, they are disposed. Intermediate filaments. Central rod-like structures and the N-terminal and C-terminal side, and their 4 subunits come together and they form this type of structures. And they bind with neighboring structures and form these strong filaments, intermediate filaments. And they are present in many cells, and different classes. They are divided into different classes. In the skin, suppose you find this keratin. Mesodermal tissue, desmin. Vimentin group. Neurofilament. Lamins. Roughly 4 groups. And it is the epithelial element, you will find keratin. Alpha and beta subunits are there. They are present in the skin, in the hair, nails, the horns and hoof of these animals. They are keratins. Alpha, and they also form polymers. And they are very strong. নখের শক্তি প্রচুর আছে। সার একটা গুতা দিলে তবে বোঝা যায় যে একেবারে সারের গুতা কখনো খেয়েছে সে জানে যে কিরকম লাগে, বুঝতে পেরেছো? কাজেই দেয়ার ইজ ইম্পর্টেন্ট স্ট্রাকচারস, কেরাটিনস, the skin, hair, nails, they are keratin. And vimentin mesodermal structures, mesenchymes, type, vimentin type, it includes vimentin, desmin, present in the muscles, glial, fibrillar, acid proteins, and peripherins in the nervous system. These are mesodermal structures, and glial fibrillary acid protein, they may be derived from the mesenchyme, and they are present in the glial cells, and peripherins it is also in the neurons. This classified in this way. Another is neurofilaments. Neurofilaments are there within the nerves. A nerve cell may be 1 meter long. Cell body may be within the spinal cord, and it can extend through and through up to the level of the foot, more than 1 meter long. And their structure, how the structure is maintained, and by these filaments, intermediate filaments structures. And they have different varieties according to their molecular weight, light, medium, and intermediate in different types of or heavy, intermediate or heavy, and this type of the molecular weight that can vary. They commonly 50 to 60 kilodalton is their dimension. And lamins are present within the nuclear membrane. Nuclear membrane. Lamins, suppose within a cell, it is cell and it is the nucleus. Within the nucleus, and it is like this, you know, nuclear pore, and at the internal side they are disposed, lamins. And by other intermediate filaments, they are connected to the cytoplasm, cell membrane. And that line the cell membrane, nuclear membrane, they are called lamins. Lamins. There are varieties A/B, lamins, intermediate filaments. And in this case, whenever the cell dies or apoptosis, then these structures are all destroyed by enzymes. And these lamins can have mutations. Lamin A has a mutation. Lamin A mutation can lead to early aging or progeria syndrome. These are the rare diseases, but for importance of the knowledge of this type. Early aging, progeria it is called. Very rare. There is, in that case, there is this lamin A is defective due to mutation. So the morphology and the function of the nucleus will be jeopardized and the cell dies. So longevity of the cell is less, and early aging and early senility will be appear in the child. These are progeria. And this nucleus is scaffolded by this intermediate cell membrane, and whenever there is apoptosis, and these enzymes will destroy this, and also so there is condensation of these nucleus and also cell together, and ultimately they are separated into different pieces and taken by the phagocytes. Now we have to discuss something about the molecular motors. Without molecular motor discussion, it is grossly incomplete. এবং তোমাদের সব বলে রাখছি, আমার জ্ঞান বুদ্ধি খুবই কম, আমি যতটুকু সম্ভব তোমাদের ইন্টারেস্ট তৈরি করার চেষ্টা করছি। যদি কোনো জায়গায় ভুলে যাই তোমরা সেগুলো বই পড়ে শিখে নেবে। ইউ অল কনসিডার সুপিরিয়র টু মি, এবং সেক্ষেত্রে ভিন্ন ভিন্ন গ্রন্থ তোমাদের সাহায্য করবে, বুঝতে পেরেছো? আর তোমাদের টেন্ডার এজ অ্যান্ড ফ্রেশ ব্রেন উইল অ্যালাউ ইউ টু লার্ন মাচ মোর, বুঝতে পেরেছো? আমরা তো এখন আমরা ডেকাডেন্ট, ক্ষয়ের মুখে। So if I can forget, then you have to learn from other different sources. Molecular motors are of two varieties mainly, kinesin and dyneins. Kinesin, dynein, and also myosin. Three types. Kinesin, it has myosin like structure, kinesin. Suppose the structure is in this way, and it is the head part, and it is the tail part, and in this way, and they are connected to in this way. And this part bear the cargo, basically. It can bind together, cargo, and it is the head part. They are all ATPases, 10 to 50 kilodalton ATPases. ATPase. And 80 nanometer, their length, and they bring the chromosomes during cell division to this apexes of this spindle, kinesin, and they carry mostly to the plus end of this microtubules, where there is assembly. And also within the neurons, throughout the cytoplasm, it can carry to the plus end neurofilaments. Along the neurofilaments and microtubule, they can move and bring the cargo. Cargo means it may be a protein, it may be synaptic vesicle, it may be mitochondria or anything else, wherever is required. And these dyneins are of this type of structure given in the text. It is the head part and it is the neck part that is embedded in this type of structures. It is the head part, only 4 nanometer. The head, neck, and this part is embedded in some proteins, and it can bear a cargo. It can bear a cargo to the minus end of this microtubules. And they are of two varieties. One is cytoplasmic variety, and another that is present within the cilia, axonal variety. Cilia, they are present in almost all the cell, and within the cilia, suppose in this case there is a cilia, and in this case if you have this section, there are tubule-like structures, 9 plus 2, their appearance. If you take the cross section of this tubular structure, then you will get at the center 2, and at the periphery there are 9 structures, 1, to 9, 1 to 9, and here there are doublets, not 3, not 3, not 3. And in this way, there are 9 doublets, 9 at the center. And in between the space, there is these dyneins, and they can help in their oscillatory. So there is movement of the cilia. And it is a unicellular element, and this movement is responsible for the movement of the cell into the through the water. But whenever they are present in the surface, and they can help to move, there is a mucus over the mucous membrane, respiratory membrane, or elsewhere. They can help to move the mucus. They are continuously active by means of their cytoplasmic dynein, and also there is axonomal dynein. And in this way, they are disposed 9 plus 2 at the center, and they are derived from the basal body. And there is a basal body from which they originate, and basal body corresponds to that of the centrosome. They have this triplet 1 through 9 position, similar to that of the chromosomes, centrosomes, they are centrosomes, and they are exchangeable. So it is really the centrosome basal body is equal to centrosomes, from which the cilia arise. But within the cytoplasm is also dynein. Now the myosins are of many varieties, 18 or more varieties. And the myosins, skeletal cell, when you learn the skeletal cell, myosin filaments are there, and they bind with the actin filament. And this myosin bears the head. By means of this movement, it causes this movement of the actin filament, power stroke, and it is an active process and requires liberation of energy by the breakdown of ATP. And over this type of these structures, there is the cytoskeleton structures. They can move. Suppose there is a microfilament like structures, and one myosin molecule is moving by means of head, it unites. This is the cargo, and it is, and they may add 1 and 2, and if there is ATP breakdown, then there is some movement of this, and in the like walking movement, they can move in this way, the 2 in this position and 1 in this position, and in this way they can move this cargo in this direction. Walking movement is possible. Like walking movement, they will move in this way. Suppose this, in case of microtubule, suppose there is a microtubule, hollow structure. And by means of a head, there is the kinesin is attached, head, and this kinesin is moving this cargo along this direction. It is formed of this alpha and beta tubules move in this direction, In this way, they have their movement, molecular movement. It may be a protein, it may be vesicle, it may be mitochondria from one cell to another. It is the negative side and it is the positive side of the microtubule. They move in this way, walking movement also. So these are these molecular motors. So molecular motors are of different types: kinesin, dynein. Dynein are 2 types: cytoplasmic dynein, and then axonomal or present in the cilia dyneins, and myosins of different types. And within the cells, it is mainly 1 to 5 type of myosin. Their dimensions do differ. In skeletal muscle, they are myosin 2, and they are elongated structures like this. But elsewhere, it may not be elongated. It may be a head and a short tail in this. Myosin 1 or so. According to the requirement, the nature produces it. And nature is always accurate. In where position, which molecular motor is efficient, which cytoskeleton element is required, and they drive this in this way. And intermediate filaments are relatively rigid structures. Intermediate filaments are rigid structures, and they, except lamins, they do not normally dissolve. During mitosis, the lamins dissolve after phosphorylation, but the intermediate relatively rigid structures, and they are not much changed. And whenever there is a tumor, and their origin can be detected by detection of the intermediate filament, de-differentiated tumor, pathologists can have some understanding from which the tumor arises with the detection of the intermediate filament of that structures, because it is relatively stable structures, and they do not disappear during cell division, except lamins. Within the nucleus it can disappear. এই মোটামুটি আমি তোমাদের একটা ধারণা দিলাম, খুব সংক্ষিপ্ত, এবং অনেক সময় অনেক পয়েন্ট বাদ রয়ে গেল। তোমরা এগুলো আলোচনা বই থেকে পড়ে নেবে, ইন্টারনেট থেকে দেখে নেবে, অনেক জিনিস আছে রিলেটেড টু, অ্যান্ড রিলেটেড ডিজিজেস আর অলসো ইউ ক্যান লার্ন।